Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. How are you today? Hello guys, let me introduce myself. Hello, I'm Ulfa Mardianti Mahmud. I'm from Biology Education University of Muhammadiyah Professor Dr. Hamka. Okay, now I want to tell you about DNA. The first, what is the DNA? Definition of DNA. First, definition of DNA. Watson and Crick proposed that DNA is made up of strands that are twice around each other to form a right-handed helix. The two DNA strands are interparallel such that the 3 end of one strand face the 5 end of the other. The 3 end of each strand has a free hydroxyl group, while the 5 end of each strand has a free phosphate group. The sugar and phosphate of the polymerase nucleotides for the backbone of the structure, whereas the nitrogenous bases are stayed inside. These nitrogenous bases on the interior of the molecule interact with each other base pairing. Analysis of the diffraction patterns of DNA has determined that there are approximately 10 base patterns in DNA. The asymmetrical spacing of the sugar-phosphate backbones generates major grooves where the backbone is forward and minor grooves where the backbone is close together. These grooves are locations where proteins can bind to DNA. The binding of these proteins can alter the structure of DNA, regulate replication, or regulate transcription of DNA into DNA function. DNA stores the information needed to build and control the cell. The transmission of this information from mother to daughter cells is called vertical gene transfer and occurs through the process of DNA replication. DNA is replicated when a cell makes a duplicate copy of its DNA. Then the cell divides, resulting in the correct distribution of one DNA copy to each resulting cell. DNA can also be enzymatically degraded and used as a source of nucleosides and nucleotides for the cells, unlike other macromolecules, and does not serve a structural role in cells. Replication of DNA. The ability of DNA to duplicate themselves with their process as follows. 1. Open hydrogen bonds. Helicase enzyme. 2. The two DNA strands separate. 3. Sum of each ribbon to form a ribbon new. Next, DNA structure. Watson and Crick proposed the double helix model of DNA. The sugar phosphate backbones are on the outside of the double helix, and purines and pyrimidines for the rungs of the DNA helix leader. The two DNA strands are antiparallel to each other. The direction of each strand is identified by numbering the carbons in each sugar molecule, and is the one where carbon is not bound to another nucleotide, and is the one where carbon is not bound to another nucleotide. Okay, next, types of DNA replication. First, conservative. The parent DNA band does not change. It is used to form the new DNA strands. Second, semi-conservative. The parent DNA strands separate and synthesis new DNA strands complement each other, resulting in a pair of parent DNA and new DNA. Third, dispersive. DNA bands stem forming segments of the segment break up, then DNA bands just are formed. Connecting sections are dotted so as to form two DNA bands just are intact. Next, RNA. The result of the transcription from DNA in virus which only had RNA alone. RNA, it has a function that is similar to the DNA. Kinds of RNA. First, ambassador RNA or RNAD or on a messenger RNA, which functions to convey genetic information code from the cell nucleus to the ribosome. Second, ribosomal RNA or rRNA, which functions as a machine assembly reader in protein synthesis that moves along the DNA. The third, transfer RNA or tRNA, which serves to transport acid amino to the ribosome in accordance with the code of the genes contained in the RNA. You can look the table difference between DNA and RNA. Okay, last, the synthesis protein. The synthesis protein is one of the process the preparation of the essential amino acid into RNA, polymers, enzymes, helicase, enzymes, hydrolase, and ATP or energy. Protein synthesis stage 1, DNA replication. A stage doubling DNA in nucleus DNA of children who format. The order is exactly to the DNA of the parent. The DNA information theory is as follows: A. In conservative theory, B. Semi-conservative theory, C. Dispersive theory. 2. Transcription occurs in the nucleus. A stage double a stage doubling DNA in nucleus. DNA children will format exactly the same with the DNA. The third translation occurs in ribosome, is the process of translating the sequence of codon on the mRNA into acid amino by tRNA. The process is as follows: A. Initiation, B. Elongation, C. Termination. Example stage of the synthesis protein: DNA antisense THG, DNA sense ATC, DNA RNAM codon UHG, RNAT or anticodon is EUC. Notes: nucleotides, phosphates, sugar base, PHB bonds, nucleoside, sugar base bonds, GB. Phosphates are always represented in terms of circle, sugar in pentacles. Okay, thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.